I'm Tracy Tomlinson. I'm the CEO of PolyBatics. Now, this will be completely different from what you've heard before, but what we have is a manufacturing platform for producing immobilized proteins on bioplastic beads. We do this in bacteria, and I'll tell you why that's even important. This is such a revolutionary technology that even the leading industry publication has featured us on the cover of the magazine. So we're really excited about the potential of this technology and where it's going. Basically, these are pictures of bacteria and beads that we grow inside of them. So it is now a platform, a true platform, because depending upon the proteins that we grow, it has a number of applications. Now I'm going to focus on one here, and what's really interesting is that the clients that we've engaged with already have seen the potential and have already contracted us to expand on some of those applications now. Things we thought we wouldn't be doing for 3 or 4 years. The main problem we're looking at is in separation, where we're addressing the cost and complexity of producing biological drugs. Those are things like Herceptin, Erbitux, all the monoclonal antibodies that Genentech produces for some of the other companies. These are very expensive drugs to produce. But what's happening is there's a trend in the market where we're going away from blockbusters towards smaller batches, more targeted patient populations. And this means that facilities that manufacture these drugs have to be very flexible as they switch from one drug to the next drug. You wouldn't want Herceptin in your Erbitux prescription. So the cleanliness requirements are quite extreme. So they're switching to flexible, disposable systems. The problem is there's no cost-effective capture step in that system. They've got bags and tubes and all that. But the actual capture step where they first harvest those drugs, there's no disposable option for that. Furthermore, there's no standard platform for the manufacturer. They go from an antibody to a different type of biological drug. So there are 3 levers that we can look at: cost, flexibility, and disposability. Now, the major pain point is in that capture I said. Using our material, we can reduce the cost of capture by 82%. Now, this allows them, instead of using the material hundreds and hundreds of times, they can use it a few times for a campaign and get rid of it. It's the use it and lose it strategy. That deals with the cost and disposability components. But because this is an engineered system, we can put different kinds of proteins, which allows that same platform to be applied to different targets, therefore giving them flexibility across a number of drug targets. The opportunity, as you can appreciate, is quite large. Now, we're dealing with not the gold of the gold miners going out there. We're providing picks and shovels. And if any of you remember the gold rush here in San Francisco, it was the guys that sold picks and shovels that made all the money in the end. So this market is currently $1.2 billion, growing to 2.8, and that's the adoption of all these flexible, disposable components, and that's the market that we're targeting. And that's been driven by, a lot has been driven by the uptake of monoclonal antibodies, and 290 of those drugs in the pipeline right now. So, oops, going the wrong way. So the reason this works in the magic of our system is the way they produce these traditional capture resins is 3 steps. They make the beads, which typically uses nasty chemicals and solvents. They grow the protein. They cross-link them with other nasty solvents, wash that up, and this is what you get. You get the proteins on the solid support, randomly oriented. With us, we take a gene, we insert the bacteria expresses that protein, and we get a bead that has all the proteins on the surface pointing the same direction. The same way every time. And because it's a plug-and-play system, what flavor do you want? And it's really that simple. So, business model, we've already got some clients. You saw those 4 ticks in the box there. We last year did more than 900 million, I wish. 900,000. We'll get there. 900,000 in revenue from some pretty popular household name companies that we're working with. We currently have 3 additional contracts with a small joint biotech and a couple of other multinational drug companies. We've got about a dozen other contracts in negotiation. Part of the challenge here is the lead times can be long. It's a year to negotiate a contract. But once you're in it, you're part of a license process, they're your customer for 15 years because it's a regulated manufacturing process. How do we make money? Well, first of all, through these services and producing these custom beads for these guys. Then after they've evaluated it, we have 2 options. We can become a supplier to them, so we have these long-term supply agreements, or they can license it and manufacture it themselves. Either way, I don't care. Actually, I prefer to have them make it. We've got a great team. I grew up down the peninsula here in Palo Alto. This is my 8th startup. I don't know why I went to New Zealand to go start another company, but whatever. Professor Barrett Ram is a world authority, literally. He wrote the book on biopolymers, and so he's the brains of the operation. I'm just the mouthpiece. And then we've got Dr. Amy Herbert, who's the VP of Process Development, who worked for ICI Biochemicals and has a lot of experience in this area as well. Board directors are all pharmaceutical executives, either currently or former. So we've got a great governance group, and we also have some of the key opinion leaders that are literally the leading authorities in their fields. We're really pleased to have that team. So we're seeking 1.275, I converted from Kiwi to US dollars. We're seeking $1.275 on this round. That's going to help us complete this last bit of process development that we need to do to scale this up so that we can supply the tons that these companies will require, and then ultimately our product launch. This is likely to lead on to a Series A early next year, so it's going to do it this year, complete some of these contracts, and then position us for a Series A next year. So that's it. I hope I didn't confuse you too much.